Welcome to BioBits and Bytes. Today we're diving headfirst into one of those revolutionary frontiers in medicine, cell therapy. I mean, imagine your own body's defense system, but supercharged and given a precise mission control. That's really what we're exploring today, how we're turning our own cells into powerful, targeted drugs.
That's a perfect way to put it. It's really about transforming our natural immune cells, the ones we already have, into these highly specialized living drugs. And they're capable of targeting diseases with just incredible precision. This isn't just about managing symptoms. It's about reprogramming the body to fight from within, using its own inherent power.
Absolutely. Today we're going on a deep dive into engineered immune cell therapies. We'll
Yeah.
be looking specifically at CR-T cells, TCR-T cells, TRNK cells, and even CR macrophages. And like you said, these aren't just treatments, they're truly living therapeutics. They have this remarkable ability to learn, adapt, and seek out specific threats.
And what's truly fascinating here, I think, is the sheer ingenuity scientists are applying to actually design these cells. They're tackling some of the toughest diseases known to us, from aggressive cancers that have historically resisted conventional treatments, tacpental complex autoimmune conditions that, well, traditional medicine often struggles with. It's really a testament to how far we've come in understanding and harnessing our own biology.
Right. So our journey today will extract the most important nuggets of knowledge and insight from a collection of cutting-edge research and clinical trial insights. We'll shed light on how these therapies work, where they really shine, and also the formidable hurdles still to overcome. Our mission, essentially, is to give you a shortcut to being exceptionally well-informed about this rapidly evolving and, frankly, deeply impactful field. Before we get too deep into the engineering side of things, maybe let's just briefly set the stage. We need to understand the core players first. We all have these incredible immune cells, our T cells, NK cells, macrophages, constantly patrolling our bodies, acting as our internal defense force. But what's truly revolutionary is how scientists are now reprogramming these natural defenders. They're using synthetic tools like CRs and TCRs, giving them unprecedented precision and power. So let's delve into these high-tech upgrades. Maybe start with how they're built and what roles these fundamental cells play naturally.
Indeed. Yeah, when we talk about engineered immune cell therapies, we're primarily focusing on a few critical types of white blood cells. These really make up the backbone of our adaptive and innate immunity. So first you have T cells. Think of these as the elite, highly specialized detectives and soldiers of the immune system. Their natural job is incredibly precise: to recognize and directly kill infected cells or abnormal cells, like cancer cells. They do this by interacting with specific markers on their surface. They're kind of like the snipers of our immune system.
Snipers, I like that. Okay, so highly targeted killers, what else?
Next up are natural killer cells, or NK cells. These are another type of lymphocyte, part of our innate immune system, the system we're born with. Imagine them as the first responders, or maybe the immediate reaction force. Unlike T cells, they don't need prior activation or specific training to identify and kill target cells. They have this innate ability to spot and eliminate abnormal cells, including some cancer cells, basically on site. They're always ready to go.
Okay, so NK cells are the rapid response team. Got it. And the last major player.
And then there are macrophages. These are sometimes called the Pac-Men, or the cleanup crew of the immune system. They are large white blood cells. Their main job is to engulf and digest cellular debris, foreign pathogens, dead cells, basically cleaning up the mess. But they're not just scavengers. They also play a vital role in initiating and orchestrating immune responses. They do this by presenting pieces of what they can engulf to T cells, essentially showing the T cells what new threats to look out for. They're both intelligence gatherers and, the heavy lifters.
That military analogy is very helpful. So T cells are commandos, NK cells are rapid response, and macrophages are cleanup and intel officers. Now, we constantly hear these acronyms, right? CRR and TCR. What do they truly represent in this context? What is a CRR, for instance? Can we break that down?
Let's break that down, yeah. CAR stands for chimeric antigen receptor. Now, this is a synthetic protein that scientists engineer directly into immune cells, typically T cells. Imagine it like giving a T cell a brand new, highly specialized antenna or maybe a custom-built GPS system. This engineered receptor allows the immune cell to directly recognize and bind to specific markers called antigens right on the surface of target cells like cancer cells. And crucially, it bypasses the body's natural, more complex antigen recognition system, the one that relies on MHC molecules. It's a direct sort of plug-and-play recognition system.
Okay, so if a T cell naturally has a built-in map, a CIR is like installing a custom navigation app. It points it directly to a very specific building, regardless of the usual road signs. That makes sense. And what about TCR? We hear about engineered TCR-T cells too, so it must be different somehow.
Precisely. TCR stands for T cell receptor. This is the natural receptor found on our T cells. In its natural state, it recognizes antigens, but only when those antigens are presented by MHC molecules on the surface of other cells. You can think of MHC molecules as little display stands. They hold up tiny protein fragments from inside the cell for the T cells to inspect. So in the context of TCR-T therapy, this natural receptor is engineered, it's made more effective, or it's redirected to specifically target particular tumor antigens that the natural TCR might not see effectively enough. So with TCR-T, we're essentially enhancing or redirecting what's already there rather than adding a completely new direct binding system like a CR.
That's a really crucial distinction. So how do these engineered cells actually see their targets in practice? What's the fundamental core difference between CR and PCR recognition beyond just the acronyms? Because this fundamental difference truly dictates what kinds of diseases they can go after, doesn't it?
It absolutely does. And understanding this is really key to appreciating their unique strengths and limitations too. For CARs, the chimeric antigen receptors, the defining characteristic is direct surface recognition. They directly recognize antigens that are expressed on the cell surface. This is vital because they don't need the antigen to be processed inside the cell and then presented by MHC molecules. They just need to physically encounter that target flag on the outside of the cell. This direct MHC independent recognition makes CARs incredibly versatile for targeting any protein that is accessible on the cell's exterior, like a unique protein marker that's abundant on a cancer cell surface.
Okay, so a CAR T cell is like that drone programmed to identify a specific logo on the outside of a building and then attack that building. It doesn't need to know what's happening inside, just that external identifier. But does this direct recognition ever create limitations? I mean, are there situations where focusing only on surface markers might not be enough?
That's a very insightful question, and it points directly to the complementary nature of TCRs. So TCRs, whether they're natural or engineered, operate on a fundamentally different principle. They recognize processed antigens. These are small peptide fragments that are presented inside MHC molecules on the cell surface. Think of it like this. A cell constantly breaks down its internal proteins into tiny pieces and then displays those pieces on its surface using MHC molecules. It's almost like showing off what's happening in its internal factory. This means TCRs can target not only surface proteins, but also, and this is very important, intracellular proteins, proteins that reside deep within the cell.
Have you ever wondered why that difference is so important? It really opens up the landscape of what these therapies can target. Because the TCRs can see what's happening inside the cell. Well, that expands the possibilities dramatically, right?
Sure. It does, and that's a staggering figure you hinted at. Intracellular proteins account for something like 90% of the cell's total proteins. So while CARs are fantastic for surface targets, TCRs unlock a much larger universe of potential targets. This includes many that are truly unique to cancer cells because they originate from mutations or abnormal processes deep inside the tumor. It's like moving from only being able to target the exterior of a fortress to being able to exploit its internal weaknesses. This vastly expands the universe of potential targets, especially when we're searching for those truly unique cancer signatures that might otherwise be hidden from surface targeting therapies. This is a massive advantage, particularly for solid tumors where distinctive surface markers are often scarce.
That's truly fascinating. That sounds like two different yet equally powerful strategies for finding the enemy. Now that we understand these fundamental differences, let's dive into the therapy that really kicked off this whole revolution and has already had a monumental impact: TCR-T cell therapy. Let's start with TCR-T cells, the therapy that truly ignited this entire field. We hear a lot about their groundbreaking successes. Where have they had their biggest impact so far? Against what specific kinds of diseases have they proven most effective?
Right. TCR-T cells have had their most significant and, well, truly trailblazing success in hematologic malignancies. So these are cancers of the blood and bone marrow. We're talking about diseases like certain aggressive lymphomas and leukemias. Their effectiveness in these areas has been, frankly, nothing short of transformative for patients. In many cases, these patients had very limited or even no other viable treatment options after traditional therapies failed. It's given hope where there was once very little.
That's incredible. Can you give us some specifics? What kind of successes are we seeing? And are these therapies actually making it through rigorous regulatory approvals to become standard treatment options?
Absolutely. The field has moved at just an astonishing pace. To date, 6 different TCR-T cell products have received approval from the US FDA. And that's for a total of 12 different indications across various blood cancers. That's a huge validation of this technology, right? It shows its consistent efficacy and safety when used appropriately. Key indications where they've shown remarkable efficacy include large B-cell lymphoma, LBCL, B-cell acute lymphoblastic leukemia, BALL, mantle cell lymphoma, and follicular lymphoma. More recently, they've also achieved approval for multiple myeloma, which is a historically difficult to treat blood cancer. These are diseases where conventional therapies often face significant limitations, and CRT cells offer a powerful new pathway to remission, sometimes even cure.
That's a significant number of approvals and indications. Can you highlight any pivotal trials or maybe specific examples that really illustrate the profound impact of their success?
Certainly. For instance, in pivotal trials, CD19 CIR therapy, which targets a protein found on B cells, actually outperformed the standard of care as a second-line therapy for large B-cell lymphoma. This wasn't just a slight improvement, it fundamentally shifted how doctors approach treatment for this aggressive cancer, establishing CIR-T as a vital new option.
Wow.
We've also seen the approval of BCMA CAR T-cell therapies for multiple myeloma, which has opened up entirely new possibilities for patients grappling with that complex disease. And beyond these, there have been high response rates reported with other CAR T-cell targets. For example, CD22 CARs have shown promise in BAL and LBCL, CD30 CARs in Hodgkin lymphoma, CD7 CARs in TAL, CD20 CARs in LBCL, and GPRC5D CARs in multiple myeloma. So it's a broad and growing success story in the realm of blood cancers, continually expanding the reach of this revolutionary approach.
Okay, let's unpack this a bit more. It sounds almost like science fiction, taking someone's cells, genetically modifying them, and then putting them back as a living drug. How do they even make these ACAR T cells? What goes into creating one of these highly specialized, incredibly potent agents?
It truly is a marvel of modern biomedical engineering. The process involves sophisticated genetic manipulation of a patient's own T cells, usually. At its core, a RT cell is a genetically modified T cell, where the CAR itself is a synthetic modular protein. You can imagine it as a custom-built tool with different functional sections, much like a Lego structure where each block serves a specific purpose.
Can you walk us through those blocks? What are the essential pieces of this Lego fire that allow it to perform its mission?
Right. The first crucial part is the extracellular antigen binding domain. This is the lock and key component. Imagine it like the unique shape of a key on the outside of the cell. It's often a specialized piece derived from an antibody, what scientists call a single-chain variable fragment, or SCFV. This is precisely what allows the CIR-T cell to directly see and bind to that specific target, that lock, on a cancer cell, without relying on the natural MHC presentation. It's the cell's new, bespoke targeting system.
So that's how it sees its target and recognizes it as an enemy. What's next in the physical structure?
Then there's the hinge region and transmembrane domain. These are often derived from proteins like CD8. Their job is basically to physically anchor the entire CRR structure into the T-cell membrane, making sure it's securely held in place. They also strategically position the antigen binding domain on the outside of the cell, ready to engage with targets. Think of it as the sturdy frame and hinge that allows the antenna to stand upright and function correctly.
Got it. And finally, what happens once it connects? What's the internal engine room you mentioned earlier that actually triggers the attack?
That's the cytoplasmic signaling domain. This is the engine room you described. When the extracellular binding domain successfully locks onto its target antigen, these internal signaling domains immediately get activated. This activation is what triggers a complex cascade of signals inside the T cell, effectively switching it into kill mode and instructing it to destroy the cancer cell. This part is absolutely critical for the cell's actual therapeutic function. It's the trigger for the immune response.
So they're not just attaching, they're also getting an internal signal to launch a full-scale attack. That makes perfect sense. Yeah. Now, you mentioned generations of CAR T cells earlier. How have these designs evolved over time? Have they become more effective, maybe like iterative software upgrades, improving performance?
That's where the software upgrades metaphor truly comes alive. Early on, the first-generation CARs were quite simple in their design. They only expressed the CD3 signaling domain, which provides the primary activation signal for the T cell to recognize and kill. While it was a foundational step, their efficacy was often limited because this single signal just wasn't enough to sustain a robust, long-lasting attack.
So they had the basic on switch, but maybe not enough power or persistence to finish the job against a determined foe, like a tumor.
Exactly. Then came the second-generation CARs, which represent the most prevalent design in current research and clinical trials today. What they did was add a crucial costimulatory domain, things like CD28 or 4-1BB, alongside the CD3. This provides a vital second signal for T-cell activation. Think of it like needing two keys to start a powerful engine, or maybe a second authentication step for a critical operation. This significantly improved the CRT cells' anti-tumor activity, making them much more potent and also enhancing their persistence in the body. That means they could last longer and continue to fight the cancer. This was a real game changer.
Right, so the second generation gave them more stamina, more power, and better overall performance. What was the next leap in this iterative design process then?
Following that, third-generation CARs were developed. These included two costimulatory domains, the idea being to achieve potentially even stronger and more sustained T-cell activation. The thinking was to amplify that power even further, though the clinical benefit over second generation has sometimes been less pronounced than initially hoped in some studies.
Okay. And then the latest evolution, these Chu-Pro-Keeks, it sound like they're not just about direct killing, but also about reshaping the entire battleground around the tumor. What exactly makes them material-opaque?
You're right. The fourth-generation CARs, often called TCR-Ts, which stands for T cells, redirected for uniform cytokine killing, took the concept a significant step further. In addition to the standard signaling and costimulatory domains, they incorporate a cytokine sequence, like for interleukin-7, IL-33, or IL-12, into their genetic programming. This means that upon antigen recognition, these TRECX don't just kill the cancer cells, they also produce and secrete these pro-inflammatory cytokines. These cytokines then act like signaling flares, further enhancing the anti-tumor effects by modulating the surrounding tumor microenvironment. They make it more hostile to the cancer and more welcoming to other beneficial immune cells. They're designed to turn a cold tumor, one the immune system ignores, into a hot one, thereby creating a more permissive environment for their own continued action and for the body's natural immune response.
So each generation is truly like a sophisticated software upgrade, making them smarter, more powerful, and even capable of influencing their surroundings. Yet, as with any truly revolutionary technology, the path isn't without its obstacles, especially as we move beyond the relatively contained environment of blood cancers and into the complex, fortified landscape of solid tumors. These powerful therapies face a whole new set of significant hurdles there. What would you say is the biggest challenge standing in their way, particularly for solid tumors?
Yeah, the biggest challenge for CRT cells, by far, lies in their limited efficacy when applied to solid tumors. While they've revolutionized hematologic malignancies, their success against solid tumors has been significantly more challenging. And this is due to several formidable hurdles that create a complex defensive system for the tumor.
I can imagine. A solid tumor is a very different beast than a blood cancer, which is more dispersed throughout the body. What makes solid tumors such a difficult target for CRT cells to successfully penetrate and eradicate?
Firstly, it's the profoundly immunosuppressive tumor microenvironment, TME. Solid tumors aren't just passive lumps of cells, they actively create a cold and hostile environment for immune cells. They are rich with powerful chemical signals. You can think of them as molecular stop signs or exhaustion commands that actively suppress the T cells. Factors like certain immunosuppressive cytokines, for example IL-6 and TGF-A, and immune checkpoints such as PD-L1 essentially put the brakes on the CRT cell's ability to fight effectively. This leads to their exhaustion, where they basically burn out before they can do their job properly.
Right.
Furthermore, conditions like a hypoxic or low oxygen TME can make it extremely hard for CRT cells to expand, survive, and remain active within the tumor.
So it's not just a physical barrier, but an active chemical and biological warfare zone designed specifically to neutralize the T cells.
Wow.
What about physically getting into the tumor in the first place?
Exactly. That's the second major hurdle. The sheer difficulty in infiltrating tumor sites. Solid tumors are often surrounded by a dense, fibrous extracellular matrix, ECM. This acts like a physical barrier, a kind of biological wall, making it incredibly hard for the larger CAR T cells to physically penetrate and navigate. Think of it like trying to fight an enemy entrenched behind thick, concrete walls. Additionally, there are often decreased adhesion molecules on the vascular walls surrounding tumors. This hinders the T cell's ability to latch on from the bloodstream and enter the tumor mass effectively. They just can't physically get to where they need to be to launch their attack.
And then even if they manage to get there and aren't exhausted, do they still have issues finding their target? It seems like a multi-layered defense system the tumor puts up.
Yes, that leads to the third major challenge. The lack of truly tumor-specific antigens, TSAs, and the problem of antigen escape. Few target antigens are exclusively found on cancer cells and nowhere else in the body. This often leads to what we call on-target, off-tumor toxicity, meaning the core T cells attack healthy tissues that also happen to express that target antigen, even at low levels, causing unwanted side effects.
Which can be really dangerous.
Exactly. Furthermore, tumors are incredibly adaptable and can evolve. They can simply stop expressing the target antigen altogether, which is known as antigen escape. This leads to a frustrating relapse even after an initial promising response. It's a constant evolutionary chess match.
That's a huge problem, undermining the very precision these therapies promise. And I've heard about some pretty severe side effects with TCR-T cells, even in the successful blood cancer treatments. Are those still a major concern, something that limits their broader application?
They absolutely are. The powerful immune response triggered by TCR-T cells, while necessary to fight cancer, can unfortunately lead to severe side effects that require careful management. The most common and potentially life-threatening is cytokine release syndrome, CRS. This is caused by the rapid, widespread release of inflammatory cytokines. These are small protein messengers that tell immune cells what to do, like IL-6, IL-8, MCP-1, and MIP-1. This cytokine storm can lead to symptoms ranging from a high fever and dangerously low blood pressure to hypoxia or low oxygen levels and even neurological changes. For instance, in a Claudin 18.2 specific CAR-T trial for gastrointestinal cancers, an astounding 94.6% of patients experienced grade 1 or 2 CRS. While often manageable with medications like tocilizumab, it requires careful monitoring in a specialized setting.
94%.
Wow.
And then there's the neurotoxicity, which sounds even more concerning for patients. What exactly is that?
Indeed. The other significant side effect is immune effector cell-associated neurotoxicity syndrome, ICANS, which encompasses various neurological toxicities. These can manifest as confusion, seizures, or even swelling in the brain. In CAR trials for B-cell acute lymphoblastic leukemia, the incidence of severe, grade 3 to 4 CRS can be around 30%, with ICANS occurring in about 10% of patients. So while incredibly effective, the balance between efficacy and safety remains a paramount concern that researchers are continuously working to mitigate and manage.
It sounds like while SCUDAR T cells are incredible, solid tumors present a whole new battleground with multiple complex defenses. So given these immense challenges, particularly with solid tumors, it begs the question, how are scientists pushing the boundaries? What advanced strategies are they developing to make these therapies not just effective, but also safer and more precise for these tougher battles? This is where the ingenuity truly shines, isn't it?
It really is. To make these therapies safer and more precise, especially for solid tumors, researchers are incorporating these fascinating logic gates inspired by synthetic biology. You can think of these like advanced security checks or if-then statements for immune cells. Instead of a simple C-target attack, we're programming in more nuanced decision-making capabilities. They might require multiple conditions to be met before an attack is launched.
So instead of just seeing target A and attacking, they can be programmed to say, if I see target A and D target B, then attack. Or maybe if I see target A, BUT, not target C, then attack. That's quite sophisticated. It's like giving them a complex decision-making tree. How do these Hindi and NOT gates actually work in practice? Can you give us some specific examples of how they're being designed? It sounds almost like building different circuits in this cellular computer.
Absolutely. Let's start with the A and D gates, which involve combinatorial antigen sensing. One elegant approach is split recognition. Here, the T cell is engineered with two separate receptors, each specific to a different antigen. For the T cell to be fully activated and launch an attack, both antigens must be engaged simultaneously. This significantly prevents on-target off-tumor toxicity if a healthy tissue expresses only one of the antigens, because the a fault, ensuring higher specificity.
Okay, so a double-check system. That enhances safety by preventing unintended attacks. What other ways are they building these ND functions? Are they pushing the boundaries of this cellular logic?
Another fascinating example is the SYNOCH system. This is a synthetic Notch receptor creating an F-then circuit. Here's how it works. Engagement of antigen A on the tumor surface triggers a genetic cascade inside the cell that leads to the transcription or production of a conventional CAR designed to target antigen B. So this ensures that the CAR against antigen B is only expressed when antigen A is also present.
Oh, I see.
This clever design prevents off-target effects if healthy tissues express only one of the targets, as the second CAR is never even produced unless the first condition is met. While it's not yet widely tested clinically, Preclinical models show its potential to prevent on-target, off-tumor toxicity when tumors and susceptible vital tissues aren't co-localized. It adds a layer of sophisticated conditional targeting.
That's quite sophisticated. It's sequential activation, almost like a biological ignition system that requires a specific sequence of events. What else are they using to build these complex logic circuits?
Well, there's also the OLI-NK platform, which stands for LAT and SLP76 fusion. In this design, key signaling molecules inside the T cell, LAT and SLP76, are each fused to an scFv that targets a unique antigen. The T cell only activates when both antigens are engaged, leading to the co-localization of LAT and SLP76, which then triggers T cell activation. This showed promise in models for R01 toxicity, which has been a concern in some monotherapy CAR-T designs. Other approaches like SuperCAR and co-label CAR also redirect specificity through protein switches that require multiple antigen engagements. They're all aimed at achieving a higher degree of tumor specificity.
That's brilliant. It's like requiring multiple passwords for access, making them incredibly precise. But what do you think is the biggest challenge with this anti-gate strategy? Does it introduce any new risks or vulnerabilities for the therapy?
You've hit on a crucial point. While greatly enhancing safety, this approach does come with a potential challenge: an increased risk of tumor escape if the tumor manages to lose expression of either one of the target antigens. If it loses one, the AD condition isn't met and the therapy might fail, allowing the tumor to continue growing unchecked. It's a trade-off between specificity and the potential for antigen loss.
That makes sense. It's a balancing act. What about the opposite logic, the ND-NOT gates, where you want to attack if you see A, but not if you also see C, which might be on a healthy cell? This sounds like an even more advanced safety mechanism.
Exactly. For N-NOT dates, also known as inhibitory CARs or ICARs, the concept is to express a prototype activating CR in trans, meaning as a separate component, along with an inhibitory CAR, ICAR. This ICAR targets an antigen that is expressed on healthy tissue but not on the tumor. So if the T cell binds to the inhibitory antigen on a healthy cell, it sends a stop signal. This prevents the T cell from activating, even if it simultaneously sees the tumor antigen. It acts as a crucial safety brake, ensuring healthy cells are spared.
So it's like a self-destruct override or a don't shoot signal,
Yeah.
protecting vital healthy tissue. That's a critical safety net, especially if the main target isn't perfectly tumor-specific.
Precisely. Examples of ICARs include those incorporating inhibitory signaling elements from well-known immune checkpoints like PD-1, CTLA-4, BTLA, and LIR-1. These are natural off switches in our immune system, just repurposed for safety here. And interestingly, the SYNOCH system we just discussed can also be adapted to function as an ND-NOT circuit. Encountering an inhibitory antigen could trigger the activation of a pro-apoptotic factor, essentially programming the T cell to self-destruct if it encounters healthy tissue it shouldn't attack. This just shows the incredible flexibility of synthetic biology in designing these cellular sentinels.
That's truly next-level thinking in genetic engineering. This raises an important question, though. Beyond just targeting, what about making these cells more robust, more able to survive and function in that harsh tumor environment we talked about? How are scientists boosting their stamina and fighting power to endure those challenging conditions?
You're touching on a critical area for solid tumor therapy. Scientists are indeed engineering SAR T cells to be more resilient and persistent. One key focus is to directly resist the immunosuppressive tumor microenvironment, TME. For instance, they can modify SAR T cells to express CD40L. This has been shown in preclinical lymphoma models to enhance IL-12 secretion by macrophages, essentially turning cold or unhelpful macrophages into active tumor-fighting allies.
Ah, interesting.
Or they can be engineered to secrete anti-CD47 antibody fragments. This helps to overcome the "don't eat me" signal that cancer cells often use. It enhances macrophage phagocytosis, getting macrophages to literally eat the cancer cells, and promotes the infiltration of M1-like pro-inflammatory macrophages into the tumor. These are ways to actively reshape the battleground in their favor.
So they're not just fighting, they're changing the rules of engagement and calling in reinforcements from within the body itself. What about their own staying power, their ability to grow in the difficult conditions of the tumor?
Right. To directly enhance their expansion and survival within the TME, researchers are incorporating things like constitutive IL-7R, interleukin-7 receptor signaling, or JAK-STAT signaling domains directly into the CAR design. This sustained signaling promotes durable tumor elimination and persistence of the CAR T cells in the body, ensuring they stick around long enough to eradicate the cancer. Think of it like giving them a continuous fuel supply and growth factors even in a harsh environment.
Yeah.
There are also novel hypoxia-stimulated CAR expression systems being developed. These enable CAR T cells to effectively expand and survive even in the low-oxygen hypoxic TMEs characteristic of many solid tumors, which typically cripple other cells. A phase 12 trial, NCT-D0568-1650, is actually investigating this for HER2 positive solid tumors right now, showing real-world progress in adapting these cells to their challenging environment.
So these are literally smarter, stronger CRT cells, built for endurance and a tougher fight, with built-in defenses against the tumor's counter strategies. What about safety? Can we put a kill switch on them if needed? Something that offers an emergency off-ramp if severe side effects arise. This seems like a critical concern when you're deploying living drugs.
That's an absolutely vital safety consideration. And yes, researchers are actively developing safety switches and controllable CARs. The concept involves incorporating suicide genes or designing regulatable CARs that can be turned on or off using small molecules. For example, inducible caspase-9, ICAS9 switches have been developed. If a patient experiences severe toxicities like uncontrolled CRS or neurotoxicity, administering a specific small molecule drug can activate this suicide gene. This leads to the rapid and targeted depletion of the CRT cells, thereby mitigating the adverse event. It's an invaluable mechanism for managing potential risks and improving the overall safety profile of these powerful therapies.
That's a critical safety net, providing a mechanism to dial down the immune response if it becomes too strong or goes off target. Okay, now let's pivot to another fascinating approach. TCR-T cell therapy. It seems to have a different set of superpowers, especially for solid tumors, because of how it sees its target. It seems to address some of the CRT's limitations we discussed.
Absolutely. The fundamental difference, as we discussed earlier, really lies in their antigen recognition mechanism. Unlike CAR-T cells that are designed to see antigens expressed only on the cell surface, TCR-T cells are engineered to recognize antigens presented inside the cell, specifically by MHC molecules on the cell's exterior. And this distinction is paramount as it dramatically broadens their target range to include a vast array of intracellular proteins.
So they can see the hidden targets, those internal abnormalities that don't necessarily manifest on the surface? What's fascinating here is that this opens up, about 90% of the cell's proteins as potential targets, whereas CARs are limited to surface markers. How does this work, and what does it mean for tackling cancer, especially those tough-to-treat solid tumors?
You've hit on the core advantage again. The mechanism is that TCR-T cells are engineered to express T-cell receptors that specifically recognize small peptide fragments. These fragments are derived from intracellular tumor proteins, meaning proteins that are mutated or aberrantly expressed within the cancer cell. The cell then processes these proteins into tiny pieces and displays them on its surface using MHC molecules. Because most cellular proteins reside inside the cell, this vastly expands the repertoire of potential targets beyond just surface markers, which, as you said, are often limited or susceptible to antigen escape.
Okay, so if a cancer cell has an internal factory flaw, producing abnormal proteins, the TCR-T cell can spot the bits of that flawed protein displayed on the outside, even if the flaw isn't a surface-level problem. This seems incredibly precise and potentially safer for healthy tissues, right? If the internal target is truly cancer-specific.
Precisely. This means a wider target range that includes truly novel cancer vulnerabilities. They can go after tumor-specific antigens, TSAs. These include unique mutations, often called neoantigens, or viral antigens like those from HPV or Epstein-Barr virus that are exclusively expressed in tumor cells. Targeting these unique internal signatures significantly reduces the risk of on-target, off-tumor toxicity against healthy tissue. They can also target cancer germline antigens, CGAs. These are aberrantly expressed in cancer cells, but naturally only found in germline cells, like those in the testes, which typically lack MHC molecules and are therefore usually not harmed. Examples include well-studied antigens like NY-ESO-1, MAGE-A1, MAGE-A3, and MAGE-4.
That sounds incredibly specific and therefore potentially safer because you're targeting unique cancer signatures.
Mm-hmm.
Are there other advantages beyond this broader and more precise target range?
Yes, there are. TCR-T cells also exhibit high antigen sensitivity. Each TCR-T cell needs only a very small number of antigens, sometimes as few as 1 to 50 presented peptide fragments for activation. This allows them to recognize even low-abundance tumor antigens, which might be missed by other therapies that require higher target density.
Right.
Furthermore, because T cells naturally possess homing abilities and are designed to infiltrate various tissues to perform immune surveillance, TCR-T cells are generally considered better suited for infiltrating the dense, complex structures of solid tumors. They can potentially overcome one of the major physical challenges faced by CRT cells in that setting. They're kind of built for tissue penetration.
That's a powerful distinction. So they can find those elusive needles in the haystack and then actually get into the haystack where other therapies might struggle.
What are some of the promising targets they're currently going after with TCR-T cell therapy? What kind of clinical progress are we seeing in the real world? Well, a prominent and well-studied target is NY-ES1. It's a highly immunogenic cancer testis antigen. It's found not only in rare cancers like synovial sarcoma and melanoma, but also aberrantly expressed in common malignancies like non-small cell lung cancer, esophageal, liver, and ovarian cancers. Clinical trials have shown significant tumor response in patients with high expression of NY-ES1. In fact, both the US FDA and the European Medicines Agency have approved an NY-ES1 TCR-T therapy for inoperable or metastatic synovial sarcoma and melanoma. That marked a major breakthrough for a therapy targeting an intracellular antigen.
So that's one of the first approved TCR-T cell therapies, a significant milestone. What other internal targets are showing promise? Are they pushing the boundaries of what's treatable?
Another significant target is MAGE-A4. This is another cancer testis antigen with high targeting potential in soft tissue sarcoma, STS, and esophageal cancer. Clinical trials, like one for STS, have demonstrated promising response rates, reaching around 44% in a challenging patient population. This indicates the broad applicability of targeting these aberrantly expressed proteins.
Okay, and what about specific oncogenic mutations, the neoantigens you mentioned earlier? Those are truly unique to a patient's cancer, right?
Yeah.
Is TCR-T therapy precise enough to go after those?
That's a very active and exciting area. For instance, the KRAS G12D mutation. This is a common oncogene mutation, present in about 30% of all tumors, and notoriously difficult to target with conventional drugs. A TCR-T cell therapy targeting this specific mutation showed a remarkable 72% reduction in metastatic pancreatic cancer in one reported patient case, with the response lasting for 6 months. This truly opens up possibilities for targeting some of the toughest to treat cancers by going after their core genetic drivers.
That's an astonishing result for pancreatic cancer. And beyond cancer itself, are there any viral targets they can go after if a virus is causing the cancer?
Yes, absolutely. HPV E6, E7 are viral antigens found in HPV-associated epithelial cancers, such as cervical, anal, and oropharyngeal cancers. These are attractive targets because they are foreign proteins exclusively found in the cancer cells. Trials in these areas have shown preliminary efficacy and high tolerability, indicating the potential for TCR-T cells to effectively address virus-driven malignancies, a unique application of this technology.
It sounds like TCR-T cells are making significant strides in targeting a distinct set of vulnerabilities. Are they also being used for other specific conditions beyond solid tumors, perhaps in blood cancers or even beyond cancer?
They are. For instance, HA1 and Wilms tumor antigen 1, WT1, are targets being explored for leukemia, especially after stem cell transplantation. HA1 TCR-T therapy has shown preliminary efficacy in recurrent leukemia, offering a potential lifeline when the disease returns. Even more strikingly, a WT1-specific TCR-T therapy called TCR-C4 achieved a 100% relapse-free survival rate in high-risk acute myeloid leukemia, AML, patients in a specific clinical context. That's an incredibly promising early signal for a challenging disease.
Here's where it gets really interesting. While TCR-T seems to overcome some of the CAR-T limitations, particularly in solid tumors and hidden targets, it has its own unique set of challenges, right? No therapy is without its hurdles, and understanding them is crucial to appreciating the ongoing scientific breakthroughs. What are the specific difficulties with TCR-T?
You're absolutely correct. Despite its distinct advantages, TCR-T cell therapy faces its own unique set of challenges. A major one is the issue of TCR mismatch and hybrid dimers. When you introduce exogenous or new TCRs into a patient's T cells, they can sometimes form these mixed TCR dimers by incorrectly pairing with the patient's own natural TCRs. These hybrid TCRs can have unpredictable specificities, potentially leading to off-target toxicity, where they attack healthy self-antigens. Or they could even cause something like graft versus host disease, GVHD, which is a serious complication where the donor cells attack the recipient's body. It's like putting two different types of keys on the same keyring and accidentally unlocking the wrong door.
So the engineered cells could essentially become confused and attack the wrong targets due to this mispairing. That sounds like a significant safety concern that needs careful engineering to avoid. What about the practicalities of making these highly personalized therapies must be incredibly complex?
That leads directly to the challenge of manufacturing complexity and cost. TCR-T therapy is inherently a highly personalized immunotherapy. It involves isolating a patient's own T cells, a process called apheresis, genetically modifying them with a specific TCR using viral vectors, expanding them dramatically outside the body in specialized bioreactors, all under extremely stringent good manufacturing practice GMP conditions, and then reinfusing them. This is a complex, multi-step production process that requires highly specialized facilities, immense expertise, and rigorous quality control. All of this leads to a very high cost, often in the hundreds of thousands of dollars, and significant logistical hurdles.
And what about the persistence of these cells once they're in the patient? Do they stick around long enough to do their job, or do they sometimes fade away too quickly? That was an issue with early CRT cells, too.
That's another area that's been a challenge in some early trials. They sometimes showed limited persistence in vivo, meaning the infused cells didn't remain active or numerous enough in the body for a sustained period to provide long-term therapeutic benefit. If the cells don't persist, the cancer can return. Ensuring they survive, thrive, and maintain their anti-tumor function in the long term is a critical area of ongoing research.
And finally, a side effect we've heard about with CRT cells, the cytokine storm. Is that still a concern with TCR-T therapies, even though they have a different mechanism of action?
Well, while it generally seems to have a lower incidence and often less severity than with CAR T cells, cytokine release syndrome, CRS, can still occur with TCR T cell therapy. This might happen especially with high antigen expression or a very robust immune response. For example, there was a reported case of a patient with fallopian tube cancer receiving MAGE-A4 TCR T therapy who experienced both CRS and ICANS, which required treatment. So while potentially less frequent, it's still a risk that needs careful monitoring and management by clinicians.
So it's a different set of obstacles, but still significant ones that demand continuous innovation.
Yeah.
What are the emerging solutions to these challenges? It feels like a scientific arms race to perfect these living therapies, constantly pushing the boundaries of what's possible, learning from each challenge.
Indeed, the scientific community is heavily invested in overcoming these hurdles, viewing them as opportunities for even more sophisticated engineering. For TCR-T cells, a major focus is directly on engineering to reduce mismatch. Strategies involve modifying the TCR alpha and beta chains themselves. This can mean subtly altering their nucleotide sequences or introducing parts of mouse constant regions into human TCRs to favor correct pairing, or even adding extra disulfide bonds between the alpha and beta chains to enhance the stability and pairing efficiency of the introduced TCR.
Okay.
These modifications are designed to make the introduced TCRs stickier to themselves and less likely to incorrectly pair with the patient's endogenous TCRs, reducing the risk of off-target toxicity. Another elegant approach is to use single-chain TCRs, which by their nature minimize mispairing by creating one continuous protein.
So essentially making the introduced TCRs self-loving and less prone to confusing their targets. That's incredibly clever molecular engineering. What about the broader strategies, like combining these therapies with other treatments to enhance their power and overcome tumor resistance?
That's a key direction, and combination therapies are showing significant promise. One powerful combination is with immune checkpoint blockade. Using drugs like PD-1, PD-L1 inhibitors, such as pembrolizumab or atezolizumab, these drugs can restore T-cell activity in the immunosuppressive tumor microenvironment. They essentially take the brakes off the T-cells that the tumor itself puts on to evade the immune system.
Right.
What's truly innovative is that some TCR-T cells are now being engineered to contain a PD-1-41BB co-stimulatory switch protein. This remarkable protein converts the normally inhibitory signals received from PD-1 engagement on the T-cell into activating signals. It effectively turns an enemy signal into a boost for the T-cell. It's like turning the tumor's defensive weapon against itself.
That's truly turning the tables on the tumor's defense mechanisms. What about using vaccines alongside these engineered cells? Can they help rally the body's own forces and broaden the attack?
Yes, vaccines are another promising avenue, particularly those customized to a patient's tumor. These customized vaccines, especially those targeting neoantigens, those unique tumor mutations or cancer testis antigens, can enhance antigen-specific T cells in vivo, meaning directly within the patient's body. This not only boosts the number of therapeutic T cells, but also triggers the activation of the endogenous immune system, promoting what's called antigen spreading. This means the host's own T cells start recognizing additional tumor antigens beyond the one initially targeted by the TCR-T cells, which is crucial for preventing antigen-negative escape by the tumor.
Ah, I see.
For example, combining NY-ESO-1 TCR2 therapy with a lymph node-targeting nanoparticulate peptide vaccine has shown promising efficacy in soft tissue sarcoma, highlighting the synergy.
So it's a powerful one-two punch. Engineered cells for direct, precise attack and vaccines to rally the body's own forces and broaden the immune response. Is there anything else they're combining these with to make the tumor microenvironment more hospitable to attack?
Yes, oncolytic viruses, OVs, are also being explored. These are viruses that are genetically engineered to preferentially infect and lese or destroy tumor cells while leaving healthy cells unharmed. OVs can convert cold tumors, those typically ignored by the immune system, into hot ones, making them more amenable to T-cell therapies. They also inhibit tumor immune evasion mechanisms and show synergistic effects when combined with T-cell therapies. They effectively create a more inflammatory and immune-friendly environment within the tumor, breaking down its defenses from within.
Those are truly cutting-edge approaches, demonstrating incredible ingenuity. Okay, let's pause and simplify that part again. We've talked CAR-T, TCR-T, now CAR-NK cells. It sounds like they might offer some distinct advantages over CAR-T, especially when thinking about safety and accessibility. What makes them so promising, particularly for these off-the-shelf treatments?
Absolutely. CR-NK cells have several key benefits that make them incredibly attractive, especially for broader patient access and safety. They address some of the core limitations of CR-T cells. First, they retain their intrinsic tumor recognition capabilities. Unlike T cells, NK cells naturally recognize and kill tumor cells without needing prior sensitization or MHC presentation. This dual mechanism, the CR-directed targeting plus their natural abilities to spot abnormalities, reduces the probability of tumor escape because they have a built-in backup system for detection.
So they have a naturally aggressive nature and a built-in backup system to ensure they don't miss targets. What about safety compared to CAR-T cells? We know CAR-T can cause some serious side effects like CRS and ICANS.
That's a huge advantage and a primary driver for their development. CR-NK cells exhibit a significantly lower incidence of severe side effects like cytokine release syndrome, CRS, and immune effector cell-associated neurotoxicity syndrome, ICANS, compared to CAR-T cells. This makes them inherently safer, which is crucial for widespread adoption and potentially even outpatient treatment possibilities. Patients typically experience fewer and milder systemic inflammatory responses.
And the off-the-shelf aspect, that's what really excites people about accessibility because it sounds like it could dramatically simplify treatment logistics.
Precisely. They offer the potential for truly allogeneic or donor-derived off-the-shelf products. This is revolutionary because they don't require strict HLA matching between donor and recipient, which is a major time-consuming hurdle for T-cell therapies. Crucially, they also have no potential to cause graft versus host disease, GVHD, a serious complication seen with mismatched T cells, because NK cells don't generate the same sustained immune memory that causes GVHD. This means they can be manufactured in advance from healthy donors, stored, and then thawed and infused as soon as a patient needs them. It avoids the lengthy and costly patient-specific manufacturing process entirely. It's a potential game changer for speed and availability.
That's a huge step forward for accessibility. Tell us about their current real-world uses or breakthroughs. Are they in trials, and how are they performing in actual patients?
They are indeed in active clinical investigation and showing very promising early results. A notable example is a phase I trial, NCT05008575, focusing on CD33-targeted core NK cells for relapsed refractory acute myeloid leukemia, AML. CD33 is a common target in AML. In this trial with 10 patients, there were no grade 3 to 4 adverse events observed except for some bone marrow suppression that resolved quickly. Only one patient experienced grade 2 CRS, which is remarkably low compared to what's often seen with CAR-T. And the results for efficacy. 6 out of 10 patients achieved MRD negative complete remission by day 28, indicating a powerful and rapid therapeutic effect. That's a very promising initial signal for a difficult-to-treat blood cancer.
Those are fantastic results for AML patients, especially with that improved safety profile. What about other blood cancers where CAR-T cells have already made an impact? Can CAR-NK compete there too?
Yes, in lymphoma and chronic lymphocytic leukemia, CLL, umbilical cord blood-derived anti-CD19 CAR-NK cells have shown significant efficacy. In one study, 8 of 11 patients responded, and 7 of those had complete tumor suppression. What's even more striking is that this was achieved without any incidence of CRS or neurotoxicity, again demonstrating their favorable safety profile in these indications.
Hmm.
The source of these cells is also a key factor for accessibility. Cord blood donations are a rich source. They can potentially yield hundreds of doses from a single donation, making them a truly off-the-shelf product readily available for multiple patients, scaling up potential treatments significantly.
And then there's ERM, another fascinating new player on this therapeutic stage. We've talked CAR-T and CAR-NK, but CAR macrophages are emerging as something quite unique. What do you think is their distinct potential, especially for those challenging solid tumors, which have proven so difficult for other cell therapies?
CAR macrophages, or CAR-RNM, are truly promising for solid tumors for several compelling reasons. They offer a completely different approach. Their most significant advantage, perhaps, is their superior infiltration capability. Macrophages are naturally programmed to migrate deeply into various tissues, including dense tumor sites, far better than T cells usually can. They're like the immune system's natural infiltrators, able to navigate the dense, fibrous tumor microenvironment with greater ease, potentially breaking through physical barriers that impede other immune cells.
So they can physically get to places T cells struggle with, but once they're in, what can they actually do that's different or maybe better? They're not just passively entering the tumor, are they?
No, not at all. Once inside the tumor, they exhibit a wonderfully broader range of anti-tumor mechanisms. They aren't just direct killers like T cells. They can literally phagocytose or eat cancer cells, directly engulfing them and clearing cellular debris. Beyond that, they act as powerful antigen-presenting cells. This means they can process tumor components and then present tumor antigens to activate the body's own T cells, essentially calling in reinforcements and instructing them on what to target.
Okay.
They also secrete matrix metalloproteinases, MMPs. These are enzymes that can break down the dense extracellular matrix of tumors, effectively softening the tumor's physical defenses and helping other immune cells, like T cells, infiltrate more easily. Plus, they can be reprogrammed to a pro-inflammatory M1-like phenotype, which is a tumor-hostile state, further enhancing the anti-tumor response.
Wow, so they're like the special forces. They can break through defenses and call in reinforcements and clear the way for them. That's a multi-pronged attack strategy built into one cell type. What's the status of CRM in clinical trials right now? Is this still largely theoretical, or are we seeing real-world applications starting?
It's still very nascent, but moving forward with exciting speed. Currently, there's only one clinical trial, NCT-0466-0929, ongoing globally for CR macrophage therapy. It's a phase 1 study in patients with solid tumors. Preliminary results from that trial, involving just 2 subjects initially, showed no significant organ toxicities, which is a great start for any new cell therapy platform. One subject did experience a grade 2 cytokine release syndrome, but it resolved quickly on the same day, indicating a potentially manageable safety profile in these early stages.
So, very early days, but encouraging on the safety front at least. What about their promise in preclinical models? Does the lab work strongly support all this exciting potential for CRM?
Absolutely. Preclinical models have shown CIRM sensitizing HER2 plus solid tumors to PD-1 blockade, meaning they make other powerful immunotherapies work even better together. They've demonstrated potent cytotoxicity, robust activation of T cells, and a significant ability to remodel the tumor microenvironment. This remodeling includes increasing the density of both CD4 plus and CD8 plus T cells within tumors, confirming their ability to transform the immunosuppressive TME into an immunostimulatory one. This is exactly what we need for effective solid tumor treatment, as they act as both direct attackers and powerful immune modulators, creating a more favorable battlefield for the entire immune system.
That's incredibly encouraging, even if it's early. It feels like these therapies are constantly evolving, with new players and new tricks emerging all the time. But bringing any living drug from the lab to patients must come with its own set of significant challenges, right? What are the broader hurdles in manufacturing and getting these highly complex therapies from scientific breakthrough to widespread patient access?
Yes, the manufacturing process itself is a significant hurdle. It truly distinguishes these from traditional pharmaceuticals. These are not pills, they're living cells, and their production is incredibly complex and precise. It involves isolating cells from the patient or a donor, genetically modifying them, often using sophisticated viral vectors to insert the CIR or TCR gene, and then expanding them ex vivo, meaning outside the body, in specialized bioreactors under extremely stringent, good manufacturing practice GMP conditions. This is followed by rigorous quality control testing at multiple steps to ensure safety, purity, and potency. Each batch is essentially a unique, living biological product.
That sounds like a long, intricate, and highly controlled pipeline. Does that translate directly to high costs and significant logistical challenges for patients and healthcare systems?
It absolutely does. This specialized, labor-intensive process requires immense expertise, highly specialized equipment, and bespoke, sterile facilities. All of this contributes directly to their inherent high costs. A single CRT cell treatment can cost hundreds of thousands of dollars. Beyond cost, there are significant logistical challenges. The patient's cells need to be collected, shipped to a manufacturing site, processed, and then shipped back, all while maintaining cell viability.
Right.
Delays in manufacturing are also a real concern. This can necessitate what's called bridging therapy for patients while they wait for their personalized cell product to be ready, adding further complexity and cost. The FDA also highlights that ensuring quality control, guaranteeing no contamination, and verifying the identity, purity, strength, and stability of the product is crucial throughout every multi-step process. That adds significant time and resources.
So, given these complexities and the rapid pace of innovation, what does this all mean for the future? How do we maximize the potential of these incredible therapies and truly bring them to more patients, making them more accessible and effective? What's the long-term outlook for this revolution in medicine?
Well, the future of these therapies likely lies in several key interconnected directions. Firstly, combinatorial approaches are paramount. This means combining these engineered cells with other immunotherapies, such as checkpoint inhibitors, customized neoantigen vaccines, or oncolytic viruses. The goal is to create a multi-pronged attack to overcome the complex resistance mechanisms tumors develop and significantly enhance efficacy, particularly for solid tumors, where a single approach is often insufficient. It's about designing a more comprehensive, synergistic strategy.
So a true multi-front attack, leveraging different mechanisms to overwhelm the tumor's defenses. What about making these therapies more widely available, moving beyond the individual, custom-made product model that currently dominates?
That brings us to the crucial push for allogeneic or off-the-shelf therapies. Developing donor-derived cells, like the cord blood-derived CR-NK cells we discussed, or even gene-edited universal T cells. This circumvents the patient-specific manufacturing and logistical bottlenecks entirely. This could dramatically reduce costs, shorten treatment timelines, and make these powerful treatments much more accessible, scalable, and readily available for patients globally, much like a conventional pharmaceutical.
And getting even more futuristic, maybe. Can we bypass the lab manufacturing entirely? Just deliver the genetic instructions directly into the body, engineering the cells right where they are needed.
That's indeed a major ambition: in vivo gene delivery. Exploring methods to deliver the genetic material directly into the patient's body to engineer cells inside them could drastically reduce costs and complexity. It could potentially transform cell therapy into a more outpatient-friendly procedure. Imagine a simple injection that reprograms a patient's own cells in their body, eliminating the need for complex ex vivo manufacturing facilities. While challenging, preliminary research in this area is extremely promising.
Fascinating. Now, beyond cancer, where else might these engineered cells find a home? Are there other diseases, perhaps even autoimmune conditions, that could benefit from this precise cellular reprogramming?
While cancer is indeed the primary focus and has seen the most dramatic breakthroughs, the potential is much broader for engineering immune cells. For instance, regulatory T cells, TREGs, which are the peacekeepers of the immune system, are already being explored for autoimmune diseases where the immune system mistakenly attacks healthy tissues. Conditions like multiple sclerosis, MS, type 1 diabetes, and graft versus host disease, GVHD, are all prime targets. A phase 1 study, the POLTREG trial, on TREG administration for relapsing remitting MS, showed no severe adverse events, indicating a favorable safety profile. Interestingly, intrathecal administration, where the cells are delivered directly into the spinal fluid, appeared more promising than intravenous administration for MS-SCIP. This highlights the need for tailored delivery methods for different diseases. So it really shows the incredible adaptability of cell therapy platforms beyond oncology.
So it's not just about supercharging the attack, it's also about recalibrating and restoring balance to the immune system for other conditions. That's amazing.
Exactly. And finally, and perhaps most importantly, continued research is absolutely necessary. Every new trial, whether it confirms success or highlights new challenges, contributes invaluable data. More clinical trials are required to gather sufficient data on efficacy and long-term safety across various diseases and patient populations. It's an ongoing, iterative process of discovery and refinement that pushes the boundaries of what's medically possible.
It's clear that we're truly just at the beginning of understanding the full potential of these amazing therapies. The landscape of medicine is truly being reshaped by our ability to engineer life itself, offering hope and precision in ways that were, well, unimaginable just a few decades ago. It's truly a new era of medical innovation, and it's exciting to imagine where it will take us next.
Absolutely. So, just to sum up, maybe remember the key differences in our living drugs. CAR T cells are the direct hit specialists. They're fantastic for blood cancers, but they face significant hurdles like the hostile tumor microenvironment and antigen escape when tackling solid tumors.
Right, the trailblazers.
Then TCR-T cells, they're the deep scanners. They're able to target hidden intracellular tumor antigens, making them particularly promising for solid tumors, despite their own complexities with manufacturing and potential for off-target effects.
Seeing the inside targets.
CRNK cells are the potential off-the-shelf option. They offer a broader safety profile and greater accessibility due to their intrinsic tumor recognition and lack of HLA matching requirements.
Safer and faster.
And finally, CR macrophages, the tumor remodelers. They are uniquely suited to infiltrate and reshape solid tumor microenvironments, acting as both engulfers of cancer cells and powerful activators of other immune cells, paving the way for a broader immune response.
The infiltrators and remodelers.
Yeah.
Got it. Each of these approaches represents a significant leap forward, continually pushing the boundaries of what's possible in medicine by harnessing the incredible power of our own biology. It's an exciting time to be following these advancements, and it truly makes you wonder what seemingly impossible medical challenges will engineered cell therapies tackle next.
It's a fascinating field to watch. Until next time, keep being curious and keep exploring the amazing science around us.
Thank you for joining again.